Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone, and welcome to this fifth live stream of our evolution course. And today's live stream is about the motor proteins and the paradox that they raise with regards to the theory of evolution. So I know we have not done our live stream for some time now, and maybe this will cause that many of you will see this recorded. So I'm going to do something today with you, which is that we are going to read together the article number 5 and see what we can together understand about the matter of motor proteins. I have one question that I have received from you guys. I'm going to also answer it, and then maybe if any of you would have some questions on the live chat, we'll also attend to that. All right, so first let's go to the article number 5. So let's do it this way. Yeah. So first, I hope that you all guys you are familiar with the course website. So to get the full benefit of the course, I urge you all to engage with the course through the website. We'll be doing that today together. So let's go. Here's the website. So it's findingtruthchannel.com/courses. You go here to courses. You pick is evolution true or false. All right. And then here you will have your lessons, your quizzes, and everything. So here's the overview, and then here's the curriculum. Under the curriculum, we are now in week 5, so we go to week 5 here. And on week 5, you'll have the motor proteins session, which is this time part of my interview with Dr. James Tour, where we talked about molecular machines and motor proteins. So you need to watch that one. And then you have article number 5, which is a description of the issue. I did not prepare a session for motor proteins like every time, like the previous 4 episodes, but maybe I will do anyway, maybe this week. So let's go to the article. So when you're opening, you'll find here the course contents. You can browse them. And here we have the session. Here's the article. You can pick up the article. We can minimize the side and look at it. So um, motor proteins are those engineering marvels. Why are they engineering marvels? Because they are what their name implies. They are machines, they are motors, they move inside the cell, and they literally would carry cargo around the cell. And this thing, the motor protein, kinesin, for example, is essentially um, a protein molecule complex. And just before we go ahead with reading the article, I would like really to show you what we're talking about. So I will show you now a small clip about these kinesins walking, literally walking in the human cell. So let's have a glimpse at that. This is a kinesin. It is this thing, this tall thing with the feet, and it has this huge cargo that is it is hanging to with these two hands it has there. They call it the tail. They look like hands to me. And then its feet are walking. Let's have a look at that again. Okay, so here it is. Those are the small things are the feet, and sometimes they call them the heads. And there's this vesicle, this huge cargo that it is carrying. You can notice the huge difference between it and its cargo. And it's moving on these rails that are called microtubules. And we are now going to understand more about that by going through uh, Article 5. So let's now go through Article 5 to understand how such a thing um, uh, does this function, and to wonder and ponder together if it makes any sense that somebody would think that this is a result of evolution, meaning it is the result of a sequence of mutations to the genetic material. And this engineering marvel will just pop into existence. So, since this is made of proteins, let's first remember together that finding the proper protein, a protein that has any beneficial use, is like looking for a needle in a haystack. Actually, finding a needle in a haystack is much, much more easier than this. Um, why? Because we have even actually discussed before the difficulty of this um, um, in previous episodes. Yet let's have a look at it. Um, yeah, let's have a look at it. So yeah, here is the um, what we have talked about before. Darwinian evolution literature talks a lot about the creation of new proteins due to random mutations. And we have discussed before that if you look at a typical protein, 150 amino acids, peptide chain protein, there are so many combinations because you have the 20 different amino acids, and so you have it at each place. There can be any one of 20 of them, and those are coded by codons, which are three-letter codes. Each letter can have one of the four different nucleotide bases. And if you do the calculation, you will find that um, there are an immensely huge number of possible combinations for this protein. Because every change in an amino acid, one of those peptides, will change the 3D structure of the protein and hence will change the function. So each one of those 150 combinations is a different protein. And sometimes just one difference can, for example, cause very serious dysfunctionality to the protein. Like, for example, sickle cell disease is caused by a single point mutation, which has a very serious effect on the hemoglobin molecule protein complex. And it causes this case or this syndrome where people will suffer from lack of oxygen because hemoglobin is not doing its job. And I'm quoting here in Article 5 this scientific paper that says that there is 1 in every 10 to the power 77 possible combinations. Only one of them will give you a protein that is functional, that has a beneficial use, that will properly fold, and then that will be usable in the living cell. Um, yeah, and some might dispute that, but we are looking at a huge search space. Um, so I'm saying we'll need to start to appreciate how those molecules can produce very complex functions on the molecular level, where those functions do not just arise from the development of new protein molecules, but even from the 3D shape to which this molecule folds, and from the interactions of this molecule with other proteins and components to form systems that deliver specific functions in a way that is quite inconceivable to the product of a random process. And this is why I picked this matter of motor proteins to be one of the topics that we talk about in this evolution course. Because you're not just looking at a protein molecule, you are looking at a set of molecules. You have the coil, you have the 2 feet that we have seen walking, We have the two necks that connect those two heads or feet to the coil. And then you have those hands that were carrying the cargo. And guess what? Those hands are cargo specific. So this kind of cargo will need this, sometimes they call them tails. They have adapters that would bind to the right cargo. So there is this integration, engineering integration between the kind of motor and the cargo that it carries, just like when we think of um, uh, cargo on trucks in cities. And then the roads themselves, or the railroads on which these motors will move, if this is a heavy truck, it will it cannot, for example, drive on sand. It will sink. So those things just don't just move around anywhere. They walk on structures like microtubules or actin, and the process of the walking that we have seen, I'll show it to you again. The very process of the walking is a series of chemical reactions. Let's have a look at it again. So this is the walking, and this walking is not just as simple as that. This is a molecule. This is an inanimate. It is just a thing. It is a molecule. It doesn't think. However, it's walking, and it's walking from target to destination, and this is because there is polarity for this microtubule. And this walking process is a series of 4 specific chemical reactions involving ATP, hydrolysis, and binding and disbinding of those two heads with the microtubules. It is this sequence: bind, unbind, move forward, bind again, unbind, move forward, bind again, that causes this movement. So you are looking at a system that has the cargo as one element, the adapter that attaches to the cargo, the coil, the two heads or the 2 feet of the kinesin motor, and the microtubule, all together in a system that delivers this function. And as an engineer, when you look at this kind of thing, just considering that this thing could happen through random mutations and natural selection is unfathomable. Yeah, so in the article, I'm also referring to what I'm calling the distribution problem. So I'm having some comments from Nestle. How are you doing, Nestle? And I'm going to get back to your comments at the end. The distribution problem. Let's imagine you are in a city. Why do we have kinesins? Cells are like those huge metropolises. And there are machinery and factories in some places in the cell, and byproducts and garbage and materials and finished products that need to be moved around. Sometimes those products are pretty big, like the kinesin that we have seen. And the cell does not just depend on chance to have those reach their destination. So the kinesins are doing, and the dyneins, other kinds of motors, are doing those marvelous functions of transporting things to the right place in the right time in the cell. And this is just mind blowing. And this is a recent discovery, actually. You will typically not hear about that as a popular thing. And I have given you in the supplementary material on the course page. If you go there, you go to the supplementary material, you will find that I have quoted there videos from the person who discovered this. He's a guy called Dr. Ron Valley, I think it's pronounced. And I have put there a 4-part series where he is actually a 3-part series where he is describing the molecular motor proteins, and an additional video also from Dewberry, which is wonderful, about their role in Kinetico and cell division. So please do look that up to see the level of complexity and integration and engineering we are looking at. Yeah, so I'm saying here it is striking to know that the cell has those robots that move around distributing materials and products around the cell, and they are called motor proteins. And I'm pointing you to look at the supplementary things. Now, how do we know that something is designed? So I'm giving here an example with this, I don't know, coffee machine and a coffee machine. So you have where you put the milk or you put the water and where you put the coffee and steam and etc. And it will just give you your coffee extract. And it is just when you see this versus a piece of rock or some rocks that have dropped on top of each other spontaneously, you will immediately recognize that one of them cannot be the result of chance, while the other one can be the result of chance. And if you find that coffee machine sitting in a coffee store, then thinking that it is chance or that it has developed there by chance over time is not reasonable. It's not a rational position. So when you find a cell where the function of transportation is needed, and you find the rails where the motor will move on, and you find the motor, and you find the adapters, and then you think that this has just come by chance, I think you need to review your position. And okay, some will say evolution as if this solves anything. But when you trace back the existence of those kinesins, for example, you find that they are available primarily in eukaryotes. So when you have cells that are big and complex and transportation is critical, you find things like the Golgi apparatus, and you find complex structures, and you find many types of vesicles and plenty of cargo that need to move around. Then you find sophistication in the microtubules, and you find the motors that are proper to the function, and they just suddenly appear in life with eukaryotes. You cannot trace them back with any proper comparable level of complexity beyond that. So raises a very big question, and there are families upon families. There are three major families, and below them there are so many species of those motor proteins, each good for something. Some of them even have directionality. So this one can go from the outer part, outer rim of the cell to the inner part, and the other one can go from the inner to the outer part, depending on the polarity of the microtubule, for example, that they are moving on. So very, very intricate design. And we're just scratching the surface with this. Here I'm showing this adapter thing. So here is, for example, this cargo vesicle, and you will have, here's the kinesin, and here's the chain, and then there is the adapter protein, the yellow one, that will cause the kinesin to attach to proper cargo. And as I was just saying, it's like a railroad, and there is trains that go this way and trains that go the other way. So kinesins, for example, will go towards the plus end of the microtubule, while dyneins might go to the minus end. And even recently, it has been discovered that some of them can actually switch gear and go on reverse. I'm also putting all this information to you on the course website. And then this amazing motion that we have seen is a result of the sequence of the chemical reactions. How do those chemical reactions happen? They happen because there is an integration between the ATP cycle. ATP is like the common fuel used in the cell, and with the heads or the feet of the kinesin motor and the microtubule. They work together to deliver this. And I have this nice video that I can also show you. It has also voice, so I cannot speak while it is being displayed. But let's have a look at it.
The motor protein kinesin is a dimer with two identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coiled coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
It's just amazing. So the elements and the engineering ingenuity of this just doesn't stop. It's not only the matter that you have the kinesin motor and then you have the microtubule, which is it's a complete fit with it. You see the head, it can bind to the microtubule and then unbind, but it is also integrated with the ATP hydrolysis process that can cause the consumption of energy inside the cell. And that those necks are there exactly arranged in a way so that the hydrolysis causes the binding and unbinding of the feet in this coordinated choreographed move, according to the expression used by Ronval himself, so that you have this exact walking process. So what we see on the very micro level inside the cell, inside the transportation system of the cell, looks like a human being who is walking. Isn't this just striking and would definitely grab anybody's attention that the design of somebody carrying something and moving on a road just looks like a human carrying something and moving on a street or something? And you think that, does it make sense? And even the process of those microtubules, when there is a cargo that wants to go from point A to point B, in many cases, we have roads on demand. We have those microtubules assembling, here I'm mentioning this, assembling and disassembling to serve the process. And this even happens in cell division, where microtubules will form those centrosomes, and then they will extend, and then they would connect to the chromosomes at a place called the Kinetico, and then this tension will be produced, and then there will be this signaling system where the motors would carry those signals that will signal that the cell is ready. And it's I also have this in the supplementary material. You need to have a look at it, the fourth video. And here is an example of how the microtubules would assemble and disassemble. I'm also putting that to you for you in the article. They would assemble this way, and then the kinesin of the dynein will move there, and then maybe after the job is done, it just disassembles. And sometimes it just keeps there because it has also other functions, like being forming the scaffolding or the skeletal structure of the cell, giving it its form. Yeah, so very specific design. This is the Kinetico I was talking to you about, and I would really like to show you this video. Let's see if I can show it to you.
This visualization isolates a single chromosome at mitosis.
So here we go.
The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions.
You see those little fibers?
The red
These are
region is the kineticore, which works as the interface between the microtubules and the chromosome. The kineticore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kineticore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension
building themselves,
which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kineticore has sensed that the tension is just right, and the chromosome is correctly positioned and attached.
See the microtubules building themselves, and
The
then
last
you'll see these
bit remaining red
dyneins.
is a stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the
See how
kineticore.
they're walking there? And it's
These
just
are molecular
a
motors
signaling
involved with
system.
the directed transport along the mitotic spindle. The orange proteins walking to the left are kinesin, molecular motors carrying protein on their back.
You see the dyneins?
Striding from the kineticore on long molecular legs are dyneins, carrying the stop signal broadcasting system.
And you see now the tension.
When all the
after
kineticores are
signaling.
ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
And here you have cytokinesis, closing cell division. It's an amazing video. Have a look at it, please. And once more, the complexity of how this thing works and the amount of integration in the system is just amazing, and the multiplicity of using elements in very innovative ways, making the design so efficient, to the extent that the microtubules that are used as rails sometimes can be used as scaffolding and sometimes can be used as tensors. They pulled those chromatids apart. And the kinesins and the dyneins, in this case, the dyneins specifically, are carrying signals. Hey, this place is now ready for separation. I'm carrying the stop signal away, so everything is green. You can now do your thing and pull it apart. And it happens in coordination with all the other chromatids giving the signals. through these kinetical mechanisms. It's completely amazing. And it says that this system is a huge engineering marvel, not just the motor, but the motor and the microtubule and the adapters and the cargo. The cargo now is a signal. And then the chromosomes arranged in chromatin, giving you these chromatids, and then this amazing structure called the kineticore. And then there are the centrosomes around it, where out of which the microtubules are radiating, and then this tension system that would split the cell apart. And if we don't have kinesins, if we don't have these systems, we cannot be living today. We cannot be speaking today, because in eukaryotes, if you don't have motors, the cell will be diseased and will die. So completely amazing. So I'm asking in the article, is it obvious enough that this is a super integrated and harmonized system design? I have to say, yes, it is. And I would really need somebody to tell me, how can this thing have arisen by random mutation? And how specifically it suddenly appears in the timeline at the last eukaryotic common ancestor, which is a hypothetical organism, of course. Yeah, so we've seen the kinetic words. I have also in the article given you some material about the same motors used in muscle contraction and relaxation, and even helping you to hear. Yeah. Very, very amazing stuff. So I would actually encourage you to read it. Now, I'm putting together a set of paradoxes. Number one, how can eukaryotes be a product of prokaryotes with all this innovation of motor proteins in eukaryotes? So motor proteins with the complexity of the system is there in eukaryotes. It's not there in prokaryotes, like bacteria. Where do they come from? I'm saying that eukaryotic life is dependent on motor proteins, which are produced by transcribing and translating eukaryotic DNA, which is on eukaryotic chromosomes that can only exist in motor proteins if motor proteins are there in the first place, because you need the kinesins to, cause the cell division of eukaryotes. So there is, the egg or the chicken there. The eukaryote cannot exist without the kinesin, and the kinesin only exists in the eukaryote. However, motor proteins, example kinesins and thymines, are not present in prokaryotes. Now, this is the first one. The second one is that motor capability with microtubules, ADP cycles, and its own cargo, this compatibility. Motors cannot function without microtubules, and microtubules are useless without motors. So if one that serves a certain function evolves without the other, it will be deemed useless and eliminated by natural selection. So how can such an integrated system fully emerge and be fixed? Noting that its amazing compatibility with the cellular chemical energy system that it uses for binding and walking. You have to notice the specificity to a variety of motor cargo on how could this cargo move around with motors. So let's imagine a piece of cargo, and it needs to be moved around by the motors, and the motor or the adapter of the motor does not exist. So how can the cargo exist without something to move it? And if the kinesin exists to move the cargo, but the cargo doesn't exist. So these things need to take place, to exist together in harmony with engineering compatibility, so that the system has any meaning. If this is just happening by random mutation, I don't know how many gazillion years of trials, not the age. I think if every atom in the universe was to give us the life of the universe, I'm not sure if this can even start to happen. So is this randomness or design? Is it more reasonable to infer that such an integrated system is the product of systems design or just chance through random mutation? So I leave you to think about this, and I'll go now to the chat and to answering the questions. So I see I have like 16 comments on the chat. Yeah, so let's courtesy to the course subscribers first answer the questions. So I had one question sent to me by email, and it says, DNA is a literal code which needs a coder. Does this go for all types of lives, even if aliens are made of totally different type of material? So yeah, interesting question. So he's saying, okay, DNA is code and a code needs a coder, but must this be the case for any living thing? Must there be always genetic material that take code? Well, it's like asking the question, regardless which type of computer, so maybe it's a PC, maybe it's a laptop, maybe it's a mobile phone, maybe it's a car with an internal computer controlling it, or a microcontroller of whatever type. Does there have to be software? Yes, you have to have software. Machines need programs. Living things need code to code for their constituents and to code for their processes. So yes, if there are aliens, I don't think it is possible to have life without the code of life. Maybe the genetic material would look different. Maybe the basis would look different. Maybe the proteins would be different, just like we see in the huge variety of life on Earth. We have DNA that is like, strands. We have circular DNA. We have different types. We have RNA, viruses. Some viruses use RNA, for example. They're not living, but they use it to store genetic code. Some have argued that there might be a different way of coding genetic information on different information-bearing molecules using, I remember, a system of 7 nucleotide bases instead of 4. But all those are details. If you have life, you need a way to organize the information that causes that life to proceed. All right, so we have plenty of comments here. Let's take them in order. So Nestlik first says, that's from Axe 2004. Yes, the paper is by Douglas Axe. It's flawed. We can discuss why you think it's flawed. The order of magnitude is huge, and I think that empirical testing of whether changing few nucleotides would cause detrimental effects to proteins or not is something that can be tested. Says it's a neat, and he's saying it's a neat animation, but that's not really how it actually works. I think he's talking about the Kinesin motor walking video. Yes, we later on in the presentation we showed how it actually works, and how it actually works is even more amazing than how it walks. So yeah, I can see how that would be an issue. Okay. And then he says most transportation inside the cell is passive. Even the motor proteins relies on the energy from Brownian motion. The motor is a Brownian ratchet that directs the random motion, unlike any machine. No, the legs or the feet or the heads of the motor protein would come to attach to the microtubule, and there's Brownian motion there, but once it attaches, the alternating movement of the feet to cause the walking effect is the result of the 4 chemical reactions that drive it. Yes, you need Brownian motion, but you need Brownian motion all over the place. Brownian motion alone will not cause that to happen. And that's why you need this further level of integration with the ATP-ADP hydrolysis cycle. The thing is, the motor proteins don't cause the motion. Yes, the motor proteins do cause the motion. Their feet alternate. It's as if you would say that human feet do not cause the motion of humans because, I don't know, there are chemical reactions in the muscles that do the motion. The whole system is a locomotive. I'm not sure what you are disputing here. Prokaryotes have actin and tubulin homologs as well. Yes, there are some actin and tubulin homologs in prokaryotes, but this is not what I disputed. I said that we don't have motor proteins there. There are traces of cytoskeletal structures in prokaryotes, but the question is about the motors, not about the actin. It's ironic that you say families of motor protein, since the term family refers to the fact that these proteins are evolutionarily related. No, it's not really ironic because I have no problem with taxonomy at all. Organizing things into families and into classifications that are useful for their study is different than saying that those families, because they go into nested hierarchies, are necessarily the result of an evolutionary mechanism. These are two different topics. So, for example, I can classify for you normal locomotives, machines that humans build, into a nested hierarchy. And it has no bearing at all on whether those have evolved from each other. So we can select some criteria and, for example, put things that move by cycling, for example, and things like that have diesel engines and things that are solar powered, etc., and things that are moving by coal, and then do further classifications to that, etc., and build a hierarchy and organize them into families and what have you. It does not mean that they have evolved one into the other. Microtubules are a good example of self-assembly. Yes, they assemble, just like the video that we have seen. It is not about the matter of self-assembly that we are looking at here. We are looking at how this design exists to the extent that part of the features of the design that you will have a radiation of a microtubule from point A to point B, where a kinesin motor would like to take that cargo from here to there. Not sure if this has grabbed your attention. It definitely has grabbed mine. It's also important to note, and I've even read some papers where there are very good hints that the very existence of the kinesin motor at attaching to the microtubule will cause continuous assembly, catalyze the assembly of the microtubule, which is pave the way for it until it reaches its destination. And then he says, oh, how nifty, therefore design is not an argument whatsoever. So when you see design, what are you going to say? Oh no, I do not want to see design. When you see design, you say that you see design. You can recognize design because design has features. And one of the very striking features of design is system integration and specificity to the function. So when you see design, you do you see design. He also says the fact that those motor proteins are reused over and over again for other processes like eukaryotic flagella and cilia is more evidence for evolution. I don't see why is that evidence for evolution at all. So you see, for example, the motors used in the kineticore example for signaling. You see them used for transporting things. You see them used in muscle motion. You see them used in hearing. And this is one of the ingenuity the ingeniousness of the design of life, where you have something that can be used for different purposes. It's called synergy. It's called reusability. He says many of these components have homologs in prokaryotes. No, there is no motor proteins in prokaryotes. Some papers on the evolution of these systems, mosaic origin of eukaryotic kineticore, tracing the origins of centrioles, cilia, and flagella, DNA. Okay, so thank you for the papers. I have gone through literature about that. Motor proteins, the way that we have shown them, are eukaryotic features. Now, if you say that, okay, I have this protein in a prokaryote that looks like another protein in a motor, I don't think that this is evidence for anything. DNA is not a code. Premise of the question is wrong already. Evolutionary literature and biology literature refers to DNA as code, so I don't know why you have a problem with that. Okay, so he says, answer to your responses. He says, no, the motion of the kinesin still relies on Brownian motion since they literally don't have the strength to pull the cargo along the microtubules. It's the Brownian ratchet. So if you ask the cargo, it will tell you that without the kinesin motor, it would not have moved from A to B. Okay. So let's give a parable to that. So you have a car, and the car moves something from A to B, but it needs to use air. Without the air, the engine will not work. So you cannot just say that the air has moved something from A to B. You need the car. Now, the car uses part of its environment, and Brownian motion is all over the place. Nested hierarchies imply common ancestry. They aren't absolute evidence for common ancestry, but common ancestry predicts it. Hence, it is evidence for it. Yes, if you have common ancestry, you would expect nested hierarchies. I agree with your statement. But you can also have nested hierarchies just because it's the same designer. Just the same example that I have given you, that because humans are the common designers, not the common ancestors of locomotives, and they have developed those locomotives over time for various functions, and whenever the function is slightly different from the function of another locomotive, they will be close to each other, but the way humans do it is not actually by starting from one locomotive and causing it to morph into another one or something. So having nested hierarchies, you cannot go from A to B there to the extent that you say that nested hierarchies proves evolution. Yes, evolution can predict nested hierarchies, but it will not be enough. It's a necessary condition that evolution is true, that you need to have nested hierarchies. That's not a sufficient condition, let's say it this way. When you see design, what are you going to say? He's quoting my statement. That's begging the question. You're assuming that there is design and then say that I see design. No, I'm saying that I'm seeing design, and then I'm asking you, when I see design, what do you expect me to say? That it's not design because I don't want to be begging the question? If you see something, you say what this thing is. He says, reuse over and over again is a feature in evolution called acceptation or evolutionary bricolage. Yes, evolution tends to hijack things that happen and consider them to be part of its features, especially when it comes to acceptation. And this is not evidence for evolution at all. It is evidence for pre-adaptation. It is evidence that there is a very efficient system of design that will cause components to have different uses. And humans, in their designs, strive so hard to make this happen because it causes a completely different order of magnitude of efficiency. No, I did not find evidence for motor proteins in prokaryotes. You insist on that. Send me something. And tubulin and actin are not the motor proteins. If you have an example for a kinesin or a dynein, for example, motor in prokaryotes, please send it to me. By the way, even if there is, if tomorrow it is discovered, proves nothing still, because how did such a complicated system happen to exist in prokaryotes anyway? A DNA molecule stores biological information. So Yoshi Birdie is saying this in the form of a genetic code made of, yes, I agree. DNA molecules are called information-bearing molecules. Same for RNA. They store information that is necessary to manufacture other things, or code for things, or regulate things. Nestig once more saying, no, they refer to genetic code as a code. The DNA itself is not the genetic code. Yeah, the DNA carries the code. I think we're discussing language now. Okay, we'll do this round, and I'm hoping to finish at 1 hour. So he says you can also have nested hierarchies because you have also nested hierarchies in human design. That's not true, and if you disagree, please conduct a phylogenetic analysis on cars. Okay. I don't know why you are saying that we cannot create classifications for things that are designed by humans, but I can tell you that this is not a very difficult task to do. You can create taxonomy for devices. Okay, take this final one. It says, when I see design quote, that's still begging the question. No, Nestle, it's not begging the question. You identify design by seeing specificity to function, by seeing integration of components to the extent of delivering the function. When you see that, you know that this is something that is designed. Okay, let's do this, the final one. We can create classifications. It says we can create classifications on design things, but you cannot conduct phylogeny analysis on design things since they aren't constricted by ancestry. I can tell you that you're begging the question here because you are assuming evolution is the one reason that you have phylogeny in phylogenetic trees in living things. And then it's more like circular. Don't you agree? So I think that we as humans will, every one of us has our biases, but if you get out of this, me and you try to get out of this and stand on a stationary frame of reference, let's say, and look at things the way they are, the very basic observation that makes us want to find an explanation is the fact that there is both similarity and variation in living things. There is also similarity and variation in human designed things. The one that we do ourselves, we know is not because of evolution, it's because of common designer. The other one, you insist it's because of common evolution because maybe somebody does not want to accept the common designer. So this might as well be the result of bias. He says that's why there is no phylogeny for cars. I don't know what phylogeny for cars will be good for. But there is classification for cars, yeah. I can guarantee that to you. So anyway, thanks everybody for attending this session. I know because we have stopped the live streams for some time, I did during that time, I did the interview with Dr. James Tour. I hope that everybody has seen it, and specifically this specific part, which I talked with him about molecular machines, and we talked about kinesins, and because he is somebody who has actually even built molecular machines like nanocars. So I can consider him an expert on the topic. And actually, some of the questions that we have gone through today, I have gone through with him. So I encourage everybody to have a look at this part 5, or even to have a look at the complete um, interview with him, which is pretty fun. And I also organized it into 8 parts. And with that, I would like to thank you very much, and see you again soon. And next will be evolution 6. We'll be talking about the protein enzyme paradox. But maybe after this experience, I will even make session 5, apart from the video interview I had with Dr. Tour. Maybe I'll even make one of our recorded sessions about the protein motors to consolidate everything that we've talked about. Thank you for being here, and see you again next Saturday.